in the world for do research with neuroendocrine cancers. So before I start, I have a question because I always this is a problem with me. Do you see the screen with one or two slides right now?
One. Your title slide. Yeah.
Okay, great. Because sometimes I have this issue that it will revert. Okay, so today I'm going to discuss about neuroendocrine tumors. I will focus first in pancreatic neuroendocrine tumors. And then if there is more time at the end, I want to talk about this new molecular neuroendocrine-like subtype that is more common in different types of cancers. Oops. Okay, cool. So, I'm going to start with the classical neuroendocrine tumors. So these are tumors that they arise from due to transformation of neuroendocrine cells. Neuroendocrine cells are diffused in different organs. For example, in lung, adrenal glands, in the pancreas, and the gastrointestinal tract. And really, the role of neuroendocrine cells is if you think about it as a communication between the endocrine system and the nervous system, and their role is to release hormones, and that's very important for the function of different organs and the physiology of the human body. Now, neuroendocrine tumors, here is the incidence. It has been increasing the past 20 years. So what you see here with orange is the incidence of neuroendocrine tumors. Is a study looking at the patients in the United States, and this one is a study here actually from Sunnybrook from Dr. Julia Hallett and Dr. Simon Singh, again looking at the incidence of neuroendocrine tumors. And you see that neuroendocrine tumors are increasing compared to the rest of the cancers. Now, it's always a bit tricky because when we look at the increase of incidence, we have to be aware that sometimes this is because now we're much better at detecting neuroendocrine cancers. And what you see below here is that, for example, lung, small intestine, rectum, and pancreas are the four major ones that are increasing over the past 10, 20 years. But what we became much better is to actually be able to detect the disease really early, so when it's still localized. But nevertheless, we still have an increase of incidence of distant metastasis, which tells us that probably there is an increased incidence of the disease overall. It's not just we're able to detect it at the early stages. Now, going back to pancreatic neuroendocrine tumors, or as we call them now, pancreatic neuroendocrine neoplasms. And this is being an evolving area where now the terminology is changing every 5 to 10 years. So for new people who enter this field is being very challenging just to follow the terminology. But with neoplasms now, we have two different types, the pancreatic neuroendocrine tumors and the pancreatic neuroendocrine cancer. For the talk today, I'm going to focus in pancreatic neuroendocrine tumors. So how do we grade these tumors right now? We grade them according to proliferation, so the Ki-67, and mainly we have 3 different grades. Grade 1, when the Ki-67 is below 3, or mitotic count. Grade 2 is between 3 and 20, and grade 3 is above 20 for the Ki-67 or mitotic count. As you see here, the pancreatic neuroendocrine tumors are well differentiated. In the grade 1 tumors, usually they are functional. For example, if it's from the beta cells from the pancreas, they will secrete insulin, and that will make it easy. Usually have early symptoms, and the surgeon will be able to excise the insulinoma, and usually these ones have very good prognosis. I'm not going to talk too much about the gene alterations today, but what I want to focus today it's this very heterogeneous population of the NETG2. which they have very, the incidence of metastasis is very variable, and their prognosis also is very variable. And I think the main challenge, and if we want to move into a precision oncology or personalized medicine, is to be able now to identify the different molecular subtypes, which might be a better way now to categorize the patients. So these are the treatments that we use, and I think Stan will talk more about this. For low grade, we use an antiangiogenic inhibitor or the M2 inhibitor, everolimus, somatostatin analogs, and peptide receptor radionuclide therapy. For the high grade, because they proliferate really fast, usually we use chemotherapy. So as I said, the idea here is that can we now identify different molecular subtypes? And I will give you an example for example, here now we have patient A and patient B, and both of these patients, what you see with yellow is a Ki67, so it seems that they have the same Ki67. But the question is now, are these patients actually have the same metastatic potential? Is it possible that one of these patients has already micrometastasis, and should we treat them in a different with different therapy? So this started back in 2009 in the group of Dr. Hanahan, during which they decided to look at the microRNA profile in the tumors. In this case, they started from a mouse model. So microRNAs, for those who are not familiar, are just a duplex of mRNA that has the ability to bind to different mRNAs and cause degradation. So that's how they regulate different signaling pathways. So what you see here is a pancreas in a mouse model, and below is the liver with metastasis. And they wanted to see whether actually all of these tumors have the same metastatic potential to the liver in this case. So here now what you see is the profile of each tumor. So each column here is a different tumor. And what you see with blue is a microRNA that is low levels, and with red is the high levels. So across what you see is the same microRNA. So for example, here some microRNAs that are low in metastasis, and some microRNAs, the same microRNAs are very high in primary tumors. So what you might expect is that now you have two different groups, metastasis and primary tumors. But in fact, when they did the analysis, what they found now is that you have some primary tumors, these 4 primary tumors here, that they actually clustered together with metastasis. So now you have primary tumors that really look like metastasis. So from here, they concluded that probably you have 2 molecular subtypes. The first one that we call islet tumors or subunoma tumors, IT, and the second one is these metastasis like primary tumors, which are the ones that have the ability to metastasize. So during my postdocs, we looked into these subtypes more, both in mouse and human, and we were able to identify different biomarkers. And here again, what I show is the mouse model. And again, you see a very early stage of progression in the case of the mouse. At 7 weeks, you have these 2 different subtypes, IT and MFP. And you see that during the progression of the primary tumor, they both grow. And at the same, at 13 weeks, they have the same size. So there is no difference in the growth in the primary tumor. But when you look at liver metastasis now, the metastasis is just from the metastasis like primary tumors. So if you have a patient that comes in at 10 weeks, you would like to know whether some of these cells are actually metastasis like primary cells, then you know that this patient at some point probably will develop metastasis. So you might want to have a follow-up more often, or follow a more aggressive therapy. So I guess what we see now, and this is not only in the case of pancreatic neuroendocrine tumors, is that cells in the primary tumor, and this is the same for breast cancer or for pancreatic adenocarcinoma, lung cancer. They develop this ability really early when they're still in the primary tumor to metastasize in different organs. So now if we go back to this scenario when we had the patient A and patient B, if we now use these molecular subtypes, now we can split this, the patients not according to Ki67, but according to the molecular features. So for example, now we have the IT, which is going to have low incidence of metastasis and good prognosis, and the metastasis like primary patients that we have high incidence of metastases and poor prognosis. So now by using its different biomarkers, here it's green and red, it doesn't really matter what it is in this case, but now you can see now we don't rely just in the Ki-67, but we have different biomarkers. So now when we go to the questions that I posed before, now we know that patient A probably is the IT subtype, it will not metastasize, probably you don't need an aggressive treatment, but in the case of patient B, that's a patient that probably will develop metastasis and you need a more aggressive treatment. What's also very important is in the case of heterogeneous populations, for example, here is a patient C, and you see that this tumor now has both subtype IT and MLP, but if you look again, the Ki-67 is very low, and probably only with Ki-67, you wouldn't know that this patient one day will relapse or will develop metastasis. So again, I think that's how we can use molecular subtypes in order to better stratify the patients with pancreatic neuroendocrine tumors. So we did look into this IT and MLP more, and I'm not going to go into all the details, but in fact, what we realized is that these MLP tumors, at least in some of the cases, they develop from the IT subtypes. So now these tumors that are indolent or not aggressive, for some reason, they have the capability to switch to these metastasis-like primary tumors, which are very, very, very aggressive and very metastatic. And again, this is now, it's a recurrent theme that we have to keep in mind, both in the clinic, but also in basic research, is this ability of cancer cells to transition from one molecular subtype to a different one, and what we refer to as phenotypic plasticity, which is now recognized as one of the hallmarks of cancers. And this is not only in the case of pancreatic neuroendocrine cancers or tumors, we also see it in, for example, in small cell lung cancer, in melanoma, in prostate cancer, in non-small cell lung cancer. And sometimes it happens spontaneously during the progression of the tumor, but sometimes it happens because of different treatments. For example, in the case of PANETs, in some of the models, really early, we saw that when we treat them with anti-angiogenic therapies, now they become metastatic. And one of the questions would be to go back and see when they become metastatic is because of this phenotypic plasticity and the switch from IT to MLP. And again, this is something that in the clinic, I think we want to be able to monitor when I have a patient that is treated for the IT subtype, does it switch to the MLP at some point? So now these are some real examples from human specimens. This was a tissue microarray. It was a collaboration with a group at the University of Bern. So what you see here on the left side, again, it's a patient that has, in this case, is the IT biomarker we use here is insulin. So you see the cytoplasmic staining for insulin. And in the third panel now, you see a patient that has this metastasis-like primary tumor. And what you see here is a nuclear staining for a transcription factor that is upregulated in the metastasis-like primary tumors. And what you see in the middle is a patient which is heterogeneous specimen now. So you see, for example, here are two cells that have this IT subtype. And surrounding here, you see these MLP-like cells. So they have the high nuclear staining. But what you see here is a few cells that actually go transition from the IT to MLP. So again, I think if we have the ability to detect this, our pathologists will be able to give more information to our clinicians, and I think it would change how we treat patients in the clinic. And again, what you see here in the case of liver metastases, all of the samples have high expression of these nuclear transcription factors, so all of them fall into this metastasis-like primary subtype. And going back again into the question is how now we use subtypes instead of Ki-67, or maybe a combination of subtypes of Ki-67 stratify patients. Again, you see now with the stage, you have increase of these metastases like primary score. This is a score of around 115 genes. But again, you see that this again is very heterogeneous. So see, even at stage 3, you have some of the samples that have very low MLP. So even these are in stage 3, you might not want to go into very aggressive treatment. And again, in stage 2, you see these ones with very high MLP. So you might want to be more aggressive, even though these tumors are not invasive yet. What was really satisfying is when we look at survival, and again, you see now the patients with low MLP score, basically all of them survive at least 5 years. But the ones with the high MLP score, they have a poor survival. Now, one of the challenges when you work with pancreatic neuroendocrine tumors is the fact that it's a very rare disease. So what we would like to do and take advantage of the samples we have here at Sunnybrook is to be able to include more patients, more samples, and maybe even develop new subtypes. So going back into, I think what we can do when we start thinking about precision oncology in the case of pancreatic neuroendocrine tumors, in general, I mean, if you think now how the different cell types in the pancreas starts from the pancreatic progenitor, what we think is that during the development of cancers, you have mutations in some of these mature cells, and then they start reverting and going back into basically to reverse, recapitulate what happens in the embryo. So now we know that actually the pancreatic neuroendocrine tumors will have at least 2 lineages, the alpha and the beta subtypes. So you see that the alpha, they have the first develops glucagonoma, the insulinomas. Then it seems that there's an intermediate stage or both of them. But as they evolve and they become more aggressive, now you have these metastases like primary tumor. And you see that these 2 lineages now, they look very similar. And in fact, what we see here is a lot of the pathways that are active in the progenitors. So it's like the cancer is pushing back in the evolution and activates pathways that are usually active during the embryonic stages. And now what we know is that in the pancreatic neuroendocrine cancer or PANNEX, the cell of origin is completely different. It's from the acinar cells. And in the case of pancreatic adenocarcinoma, it's the ductal cells. So I think when we start thinking how we treat patients, what we would like to do at the end is to be able to identify whether it's in any of these stages of the cancer progression. And then what we would like to do is to have different therapies for each stage, but also probably also preventing switch from one molecular subtype to another, so inhibiting this phenotypic plasticity. So I'm going to go back and I want to talk a little bit more about the microRNA. So here in one slide, I will just emphasize the work I did in 6 years, which was really understanding the role of microRNAs during the progression of disease. So what we found is that different microRNAs, they will regulate different capabilities of cancer. For example, we show that 181 microRNA is responsible for this switch from one subtype to another. And different microRNA, 137 now, will be responsible for increase the invasion. So you see here now in insulinoma, when you overexpress 137, these tumors now become really invasive. And then the third microRNA, the 23B cluster, now again, it will increase the metastasis. So now you see here a liver with only 1 metastasis in the case that you have this insulinoma subtype. But when you upregulate or increase the levels of 23B cluster, now this one becomes really, really metastatic. So again, the message here is that we spend, I think, in oncology 20 or 30 years talking about gene mutations and how gene mutations or oncogenes and tumor suppressors are very important for the initiation. But I think one thing we're still lacking, we haven't incorporated yet in the clinic, is how do we monitor, for example, and how to incorporate the information that is known is not because of mutations, for example, in this case, microRNAs. So if you think about here, like this cell here, which is the hyperplastic, the early tumor, and this one here that is metastatic, they have exactly the same mutations. However, what's changing during the evolution of and the progression of this cancer when it becomes invasive, metastatic, or a switch subtype is all because of microRNAs. So maybe something we have to consider is how do we use microRNAs more in the clinic. All right, so why Sunnybrook? So when I finished my postdoc and I was applying for jobs, the advice I got is that if you are not going to a site where they study neuroendocrine tumors, just don't talk about it. Because they told me such a rare disease, nobody will want to hire. And I was looking at Sunnybrook, I was like, oh my God, this is the best site to go if you want to work with pancreatic neuroendocrine tumors and in general neuroendocrine tumors. And I think it's for the people here at Sunnybrook, we know very well that this one is the biggest center in Canada. It's one of the biggest centers in North America and the world. And really what I hope that I will be able to do is to really take advantage of this. And of course, collaborate with the amazing team of clinicians and pathologists already here at Sunnybrook that work with neuroendocrine tumors. So this is a kind of where I'm trying to initiate different projects, and I would really appreciate your feedback. And that's what the different areas are. I think that would be very important to develop in order to transfer more of the knowledge from the bench to the bedside, and also probably bring some of the problems that we see in the clinic back into the lab. So the first thing we did is that we realized that there is a huge amount of blocks of tissue samples here at Sunnybrook. So what we started now is to develop this tissue microRNA from gastroenteropancreatic neuroendocrine tumors. So in collaboration with the pathologists here, we identified around 800 samples. We applied for a grant and we got funding, and now we're developing what we think is going to be the largest tissue microarray for gastroenteropancreatic neuroendocrine tumors in the world. So once this is done, what we want to do is we want to see whether microRNAs now we can use them as biomarkers, either alone or maybe with also with Ki-67 or different other genes. For example, some of the transcription factors that we found are important for this switch from IT to MT. We also we want to collaborate with the microRNA work will be done with Dr. Neil Renwick. He's at Queen's University. With Dr. Jesse Chow, he's a new investigator here at SRI as well. We want to also look at this microRNA and we want to use artificial intelligence to see whether that can also allow us to identify these different molecular subtypes. And also we want to use multiplex immunofluorescence with a group of Dr. Martin Yaffe and Dr. Alison Chang. Again, the idea here is whether we can develop a small multi-biomarker that will allow us again to identify the different subtypes. With Hon and again with Neil at Queen's, what we put as a proposal in a Terry Fox PPG grant is what we want to do is now is to develop liquid biopsies, and the idea is now is with the specific cell surface markers we'll be able to pull down EVs that secreted from neuroendocrine tumors. And what we want to look into these EVs are different microRNAs. Again, now we think that we can combine specific cell surface markers from the neuroendocrine tumors. And again, the microRNAs will be able to give us the different subtypes. The third thing area is biobanking and developing different models for patient personalized models or patient avatars. Again, with the bank. We wrote now the protocol for biomarking of solid tumors. So this is going to be a protocol that every investigator here at Sunnybrook will be able to use for biomarking of solid tumors. I'm not sure if it's been submitted in the REP yet, but the idea now is that when you have a patient, we would like to take some of the sample for biobanking, but also develop organoids and also the PDX-OVO, which is a technology that is in the group of Dr. Hong Leong. And again, this is a collaboration for the organoids. I would like to collaborate with Dr. David Andrews, and of course, Dr. Julie Hallett, Calvin Lau will be very important as a surgical oncologist, and again with Dr. Simran Singh. So once we have all these components in place, so my lab what's trying to do now is to identify different specific therapeutic vulnerabilities for the different subtypes. So we are looking at different transcription factors, what we call master regulators, which we think are very important for the switch from one subtype to another. We're also looking at FDA-approved drugs in collaboration with Dr. Hansen K. And I think once we have a good number of organoids, we can also use the small molecule libraries with a group of Dr. David Andrews. In collaboration with Dr. Bob Kerber and Chao Wang, we also want to look at immunotherapy. One of the things we identified is that when we have the switch from one subtype to another to the more metastasis like primary tumors, these tumors, they become immune invasive and they upregulate a lot of their immune blockers, for example, PD-L1. So, so this is the it's my plan and what I would like to bring in always in collaboration with clinicians and other scientists at Sunnybrook in terms of the pancreatic neuroendocrine tumors. And I have a couple of more slides since Kerber and Bob mentioned the pancreatic, the neuroendocrine-like tumors. I'm just going to introduce this subject, which is something I think it's as a clinicians and as a center here, we have to keep in mind is these neuroendocrine-like tumors. So this is the whole story started, let's say in 2019, after this study in which they showed that most of the epithelial cancers, when they become really aggressive, they will develop this neuroendocrine-like molecular subtype. And when we talk about neuroendocrine-like tumors, what they do is that they activate or they upregulate a lot of the pathways that are usually active in neurons, for example, in presynaptic process or neurotransmitters, central nervous system neuron differentiation. They also upregulate genes that are involved in cell cycle splicing, and then they downregulate the genes that are involved in immunes and adhesion molecules. And for example, what you see here, again, this is a score for the neuroendocrine-like subtype. And you see the score is very low in normal lung. You see here now it's a lung adenocarcinoma. And then again, you see that some of them will have very high score of this neuroendocrine-like molecular subtype. And these are the primary tumors that usually will metastasize. The same thing is in prostate cancer and the same thing in breast cancer. And there was another study published a year earlier before in 2018, again, in which they look at 10,000 cases. These are 32 different cancer types. And again, you see this molecular subtype here, which has the worst overall survival at this neuroendocrine-like molecular subtype. So it seems that a lot of epithelial cancers, when they become very aggressive or metastatic, they switch to this neuroendocrine-like phenotype. And this is very important. For example, I mean, I think the best examples now is resistance to targeted therapies. We know very well in prostate cancer, one of the mechanisms for resistance to anti-androgen treatment is the switch to neuroendocrine-like prostate cancer. The same thing in non-small cell lung cancer. When you treat with the EGF treatment, again, they switch to a small cell lung-like neuroendocrine cancer. So it seems that this neuroendocrine switch is involved in resistance to therapy, but also something that is new and it was brought to my attention from by Dr. Kerbel is that and actually was published, but I missed it in 2020. Is when you look at now immunotherapies, we know that we have three different types: the infiltrated tumors, in which now you see the T cells are able to infiltrate, and probably these ones will respond well to immune checkpoint inhibitors. You have the tumors in which the T cells now are excluded, are in the stroma, and this is the excluded subtype. And then you have this subtype that is called desert. So there are no T cells, and from what I was told, that even if you, let's say, use CAR T cell therapies, you can't get them into these tumors. So what they saw in 2020, actually, it's that these tumors that are desert, so you have no T cells, no infiltration at all. Again, they have this new neuroendocrine-like molecular subtype. And of course, right now, we don't know why this is happening, but I suspect that maybe it has to do something with the neurotransmitters that are secreted by these epithelial cells. Once they switch to the neuroendocrine subtype, maybe they don't allow the T cells to infiltrate. Something that is also, again, very relevant.
Can I break in, Iacovos? You're running a little bit over time. Maybe I don't think we're going to have time to do the brain metastasis part because we want to try and wrap this up around 6:30. Is that okay?
Here's the last slide. I can just quickly run through it. Just one slide. Here's the last one.
Okay.
Yeah. Again, the idea here is that if you look at different brain metastases from different epithelial cancers, again, you can see that during metastasis, these epithelial cancers, they will activate neuronal genes. So again, these neuroendocrine pathways are also involved specifically in metastasis. And I will stop here, and this is my last slide. And I want to acknowledge Safa and Ilashi, who just joined my group, and they're working in some of these projects, and everyone for coming to my talk.
Thanks.